The cell. Anton von Leeuwenhoek was the first person to observe and document living cells. Robert Hooke developed a microscope that let him study cork tissue, and he coined the term cell. Matthias Schleiden concluded that all plants are made of cells. Theodor Schwann stated that all animals are made of cells. Rudolf Virchow said, "Where a cell exists, there must be a pre-existing cell." The cell theory states that all living things are composed of cells. Cells are the basic unit of all organisms, and all cells arise from pre-existing cells. Cells are very small and are enclosed by a membrane that regulates the passage of materials. They also contain nucleic acid, which directs the cell's activities and controls inheritance. There are two types of cells, prokaryotes and eukaryotes. Prokaryotes have no nucleus or other internal membranes. All bacteria are prokaryotes. Eukaryotes have a nucleus and are more complex cells, like human and animal and plant cells. Differences between prokaryotes and eukaryotes. Prokaryotes have no membrane-bound organelles, while eukaryotes do. Prokaryotes contain a single circular chromosome, while eukaryotes are linear, containing 46 chromosomes in each nucleus. Prokaryotes can contain plasmids, eukaryotes cannot, and there are many other differences. The theory of endosymbiosis is that eukaryotic cells evolved when free-living prokaryotes took up permanent residence inside other large prokaryotic cells, which was the origin of the complex eukaryotic cell. A cell's form is dictated by its function. A nerve cell, for example, is long and spindly. Although different cell types have different functions and appearances, they all contain the same organelles. Structures of plant and animal cells. Plant and animal cells have many similarities, like ribosomes and mitochondria. However, plant cells only have cell walls and animal cells only have centrioles. The nucleus contains chromosomes made of DNA that is wrapped with special proteins called histones into a chromatin network. Chromosomes contain genes, bits of DNA that code for polypeptides. The nucleus is surrounded by a selectively permeable double membrane or envelope that contains pores and allows for the transport of large molecules such as RNA out of the nucleus and into the cytoplasm. The nucleolus is a prominent region inside the nucleus of a cell that is not dividing. Components of ribosomes are synthesized here. Nucleoli are not membrane-bound structures, but are tangles of chromatin and unfinished bits of ribosome. One or two nuclei are commonly visible in a non-dividing cell. Ribosomes are the site of protein synthesis, and ribosomes are either freely in the cytoplasm or bounded to the ER. The endoplasmic reticulum, or ER, is a system of membrane channels, and there are two types. The rough ER, which has ribosomes on it and is the site of protein synthesis and also transports, and the smooth ER It synthesizes lipids, connects rough ER to the Golgi apparatus, detoxifies the cell, and carbohydrate metabolism. The Golgi apparatus lies near the nucleus and consists of flattened sacs of membrane stacked next to each other and surrounded by vesicles. They modify, store, and package substances produced in the rough endoplasmic reticulum and then export them to other cells. The lysosome is a sac of digestive enzymes enclosed by a single membrane. It's the principal site of intracellular digestion. They help renew the cell by breaking down and recycling cell parts. Programmed cell death, called apoptosis, is critical, and plant cells do not usually have lysosomes. The mitochondria is the site of cellular respiration. A very active cell would have many of them. The mitochondria consist of an outer double membrane and a folded inner membrane called cristae. Mitochondria contain their own DNA and can self-replicate. Vacuoles are single membrane-bound structures that store substances for the cell. And they're storage. Vesicles are tiny vacuoles. Plastids have a double membrane that are found only in plants and algae. There are 3 types of plastids: chloroplasts, which are green because they contain chlorophyll and it's the site of photosynthesis, and chloroplasts have a double outer membrane. And like chloroplasts, like mitochondria, contain their own nuclear material and can self-replicate. And then another plastid is leucoplast, which stores starch, and chromoplast, which is responsible for the red, orange, yellow coloring of carrots, tomatoes, etc. Cytoskeleton is a network of protein filaments that extend throughout the cytoplasm and gives the cell its shape and enables it to move. There are 2 types of cytoskeleton: microtubules and microfilaments. Microtubules are thick, hollow tubes that make up cilia, flagella, and spindle fibers. Microfilaments are made of protein actin and help support the shape of the cell. They enable animal cells to form a cleavage furrow during cell division, amoeba to move by sending out pseudopods, and skeletal muscles to contract by sliding along myosin filaments. Centrioles and centrosomes. Centrioles and centrosomes organize the spindle fibers required for cell division. Only animal cells have centrioles and centrosomes. Plant cells have microtubule organizing regions instead. Two centrioles perpendicular to each other make up centrosomes. Centrioles and spindle fibers have the same structure. Cilia and flagella. Cilia and flagella have the same internal structure. Both are made of microtubules. The only structural difference is in the length. Cilia are short and flagella are long. The cell wall is not found in animal cells, but in plant cells. Cell walls of fungi consist of chitin, and plants and algae have cell walls made of cellulose. Cytoplasm and cytosol. Cytosol refers to the semi-liquid portion of the cytoplasm, cell or plasma membrane. The membrane is a selectively permeable membrane that controls what enters and leaves the cell. It is described as a fluid mosaic because it is made of many small particles that are able to move around in order to control what enters and leaves the cell. The plasma membrane consists of phospholipid bilayer with proteins dispersed throughout. Molecules of cholesterol are embedded within the membrane, making it less fluid and more stable. The external surface of the plasma membrane has carbohydrate chains attached to it that are important for cell-to-cell recognition. An average cell membrane consists of about 60% protein. These proteins provide a wide range of functions for the cell. Some membrane proteins, like ATP synthetase, act as an enzyme. Some, like those involved in the sodium-potassium pump, transport ions into and out of cells.